The overall goal of these clock scan protocols is to allow the fast characterization of the average pixel intensity within, along the border of, and/or on the outside of selected regions of interest.
This method can help answer key questions in tissue image analysis about the average pixel intensity within specific regions of interest. The main advantages of the clock scan technique are that it can be used to quickly characterize the average pixel intensity from or between single or multiple regions of interest.
For standard clock scan analysis, under the File menu in the appropriate ImageJ program, select Open to open an image file of interest. For example, here, a tissue section with neurons labeled for a protein of interest will be analyzed. Select an appropriate drawing tool and outline the region of interest. Select Plugins and Clock Scan from the File menu to open the standard Clock Scan Protocol pop-up option window. The Region of Interest Manager window, with the outline automatically added, will also open. In the Plugin Option window, use the scroll bars to change the X and Y coordinates of the center of the region of interest as appropriate. Depending on how much of the background region outside of the object should be covered by the scanning, use the Scan Limit scroll bar to adjust the scan limits. Select the Real Radius, Subtract Background, Polar Transform, and/or Plot with Standard Deviation checkboxes as experimentally appropriate. Then